The next presenter is Dr. Bahar Baviskar, who is a wildlife veterinarian with 20 years of experience in conservation medicine. Dr. Bahar is currently working as president of the Society for Wildlife Conservation, Education, and Research. His research is focused on diseases of free-ranging wildlife. With the One Health approach, he's involved in standardization of practices for health surveillance. And today his presentation will be about implementation of One Health with reference to study of disease prevalence. So Dr. Bahad, it's up to you now.
Thank you, Edu. And welcome you all. Namaskar, everyone. So I hope my slides are visible and I'm audible.
Yes.
Yes, great. So as already Dr. Mini has covered the topic that a veterinarian perspective in One Health and disease investigation. To move on further, the same thing I would experiences that we have faced during implementation of One Health with reference to particularly study of disease prevalence in wildlife areas. So as an organization, when we started rescue and rehabilitation work of the wild animals, we used to receive so many cases of different species from, it was more than almost 400 species, birds, reptiles, and mammals, and everything. Most of it was included as urban wildlife. But when we started doing some research with the help of veterinary colleges, institutions, and some of the disease laboratories, so we came to know that data regarding the diseases is lacking a lot. There is no detailed study which is happening. At that time, actually, there was no study in particularly was being conducted with focus on wildlife diseases. There were occasional reports and there were occasional incidences of some of the mortalities. They used to brought us these animals to pathology department, conduct the post-mortem examination, collect the samples, and make it as a confirmatory diagnosis that okay, this animal died of particular disease. But there was no compilation. I mean, there were few veterinarians who were exclusively working on animals and zoo animals, particularly reporting diseases. But with changing scenario as an organization, there was adaptive strategy from our side that we need to start working on One Health implementation when we are trying to, contribute from our side as a veterinarian. So basically, when we started thinking about One Health, and even I strongly believe that One Health approach, it is in still nascent stage. What exactly to be done on ground? How the first step should be taken? Rather, what should be the first step? And there are so many things that are broadly covered in One Health approach that it becomes rather confusing to where to start with. So basically, when we talk about One Health, it's definitely a multisectoral transdisciplinary approach, multisectoral collaborative approach that deals human, animal, and environment. So no need to talk about COVID-19, which shook the world completely, and it shook all three interfaces, human, animal, and environment, where everything was halted. So COVID is one of the example, but there are many other diseases, including rabies, including many other zoonotic diseases that are affecting. Some of the diseases are not affecting the human being, but they are affecting domestic animals and wild animals are being transmitted vice versa. So when we start dealing One Health approach, that includes number of diseases happening that are being found on the ground, in the animal, in the individual, at species level, at habitat level. There are many environmental hazards that includes climate change, flood, natural calamities, and several environmental issues that includes conflict mitigation, man-animal negative interactions, that are affecting One Health. So when we talk about One Health as a veterinarian, my specific focus is more on disease prevalence. That is what I have been doing, and trying to simulate some of the theoretical models on ground. So when we talk about disease prevalence, it would be certainly a number of cases, total number of cases that are being found in particular area in the given population at a specific time. And it is very important epidemiological matrix nowadays when we are in a position to talk about One Health implementation from veterinary perspective. So we need to understand what exactly that disease is causing the havoc, what is the burden of that disease on ecology, and what are the resources that are affecting the disease transmission and disease prevention and control and eradication strategies. So it's a holistic approach that we need to consider on a bigger picture. But when we work on the ground, I absolutely understand that bigger picture is something that looks very fantasized and romanticized idea. But when on you are ground, uh, you have to focus exclusively on one individual or one species at a time. Uh, and there are a number of parameters that will decide our groundwork. But if we talk about disease prevalence study and modeling it, so these are some of the terminologies that we should use. Point prevalence, that any occurrence of any disease at particular time at particular place. Period prevalence, that would be variation among visibility of the disease and its clinical manifestation. And another prevalence would be lifetime. There would be many diseases that will be, merging and upmerging, and again they are coming in visibility with clinical manifestation, with a lot of mortality of animals, individuals, including multi-species model. So this disease prevalence has to be studied. Unfortunately, with lack of resources, lack of funding, uh, and lack of technical and skilled manpower, many of the time we could not focus on study of disease prevalence. So there are confusions, there are biasness about implementation of disease prevalence or disease surveillance modeling, how to go with it, how whether we should start in free ranging wild animals, whether it is possible, whether it is feasible, and whether it is required. I mean, one or two mortalities in wildlife with particular disease is absolutely okay if it's going to be natural. But if the disease is spreading uh, unknowingly, affecting multi-species, and if we do not have any idea about the disease transmission, where disease sources, where it's coming from, how much is the extent of that disease, then in that case, on ground situation, it's going to be very difficult to prevent any outbreak if uh, it happens. So what we started as a surveillance system, that we were reporting some of the cases that were brought to us for postmortem, or the rescued cases which were being investigated thoroughly. And we started one active wildlife health surveillance model in TCP, which is Tiger Conservation Action Plan, uh, which is mandatory for most of the tiger reserves to be followed. So unless you change some of the things in policy, the implementation gets difficult. So what we did, we started making contact with some of the field directors of different tiger reserves in India, uh, with more focus on central India, where we started uh, implementation of wildlife health surveillance in TCP. So what it should be, how it should be done. So there was a lot of debate on it, how it should be done. So we had suggested, and based on the available literature, active and passive surveillance are the two strategies that can be effectively implemented. So we started working on buffer villages of the tiger reserves, where we knew it that there is a lot of interface between domestic animals and the wild animals. So collecting the samples from the domestic animals would be obviously a lot easier as compared to collecting it from wild animals. And there are many legal issues as well that we need to get the approval of Chief Wildlife Warden or the authority of the Forest Department to collect the samples from wild animals, especially free-ranging wild animals where they are not rescued, they are not accidentally coming in your treatment facility. So in that case, we have to rely on the passive surveillance completely. When we are getting some of the cases, in the picture is one of the Hanuman Lungur, which was found positive with rabies, and it has history of biting 20 people in around the tiger reserve. So now active surveillance includes the proactive approach where we go in the villages or even in the free-ranging wildlife, we collect the samples, and there are many sampling techniques what we have to rely on. So that we will discuss again. So when we started this designing of prevalence study, as I mentioned earlier, that active surveillance and passive surveillance was done in simultaneously phases. So that included a lot of things. I mean, when we talk about disease prevalence as a wildlife veterinarian, I am of the opinion and of the strong opinion rather that we need to understand the basic wildlife biology, the basic biology of the species that we are going to interact. So, for example, if we are talking about the raptors, and if there are some, as earlier Dr. Mina was saying, that there was some toxicity cases, including botulism. So we need to understand what these species are doing. So there are certain models that we use, like asking 5 to 6 questions. Who is the target species that is being affected, or who is that individual belong to which species? What? That includes what are the symptoms, what are the problem, what are the situations on ground that that individual or that species is facing. Where? That includes spatial distribution, whether that is being found in the water body or in the free-ranging wildlife or in the interface or in the villages or in the urban wildlife scenario. When? When it's happening, whether it's happening periodically in particular season or during the particular hours of the day. So that affects, for example, I would say there are many blood parasitic diseases which show some of the symptoms of fever in particular hours of the day. There are some diseases which are very seasonal, like avian cholera, avian botulism, echinococcus, which is found in certain areas with moist conditions. So we need to consider all these factors when we design the study. And we need to have the basic understanding of the biology of that species. So here we were collecting the samples for canine distemper and rabies and many other diseases. We used to collect it for RT-PCR for antigen testing and serum collection for antibody testing for canine distemper in some of the tiger reserves in central India. So as earlier said, again, I would emphasize again the same thing. When we start modeling of disease prevalence, when you start collecting the samples, I absolutely understand the ground situations are really challenging when we collect the samples. There are many lacunas. Many field veterinarians or in the frontline staff of the forest department or even from the animal husbandry department do not know the standardization of the sample collection for different testing. For example, if we are going to collect the samples for rabies, how to collect the samples, how to collect the brain samples. Many times it happens that we have asked the vets on ground that you just collect entire animal and just send it to lab. If it's small, that's okay. But if it's big, then it's quite challenging. You have to collect the brain tissue samples. You have to collect something that really does matter. So uh, when we talk about disease prevalence, we were saying that understanding basic biology or wildlife biology of that species, or you need to have some understanding of the habitat. So I would quote one of the example where there was mortality of the raptors in particular water body. Uh, and, and ultimately it was found that the raptors were dying of avian macular myelopathy, which is found in particularly raptors in for a very brief period of time. Uh, for your information, the disease is, causes some non-inflammatory spongy degenerative changes in the white matter of the central nervous system that was affecting the raptors and some of the birds which were eating some plant material like coots. So now if we look at the one individual and try to investigate the case on the basis of the mortality pattern, then it's quite challenging if we entirely focus on raptors, particularly eagles. So in this case, when the birds were dying, the mortality pattern was very short at very constant location. So that sample collection was very crucial, where the birds were collected, the postmortem was performed, the spongy degenerative changes were the commonest clinical manifestation, rather pathogenic lesion in all the animals, all the birds. So when investigation, we made some contact with the biologist and some of the uh, botanists. So the interaction, as we said earlier, that it should be collaborative approach, it should be multidisciplinary approach. So when it was discussed with some of the botanists, it was confirmed that it was the prime reason of the transmission was the hydrilla, because it was found that most of the leaf surface of this hydrilla was covered by the cyanobacterium, epiphytic cyanobacterium species, which caused certain nervous disorders in the birds and which were eaten by the raptors. So if you look at entire cycle, it's just a cascade of one event, and it is being followed by another. So that sample collection is very crucial. And when we are talking about prevention strategies dealing disease prevalence, then we need to understand the biotic and abiotic factors that are affecting that disease transmission. For example, some of the geese species are being affected by leukocytosome species, and the vector of this disease is black flies. So if we want to control the spread of this disease in geese, we need to understand the basic biology about the black flies, which are the carrier, which are the vector for this disease. So they they do have that foraging sites of less than 5 meters. So if we keep the birds in the cages which are more than 2 to 5 meters height, the prevalence of this leukocytosis was found to be very low. The cases were very low. So what I mean to say is that there are a number of factors that are affecting uh, the disease transmission. So while collecting the sample from the wild animals particularly, it's very crucial that we need to understand the biology of that species. Having said that, there are multiple factors affecting that sample collection as well, like for example, altitude, height, mountain, or the water bodies, presence of water bodies, or even the climate change. For example, earlier speaker was talking about the botulism. So botulism is happening in many water bodies where the birds are dying in very big number. But if we follow some of the factors and follow the history, then we will come to know that rainfall is one of the factors that is definitely affecting the cases of botulism. And not just that, there are stormy weather which has caused storms, may have increased the mortality of the birds. And as we know, mortality bird or recovery of the carcass is a very poor indicator of the wildlife mortalities that are taken into consideration. Uh, as statistical modeling, it's like not more than 1% carcasses are recovered in bigger mortality. So it's a big issue, very big issue. So sample collection, as to be done very, very, very cautiously, and there are a number of steps that has to be followed rigorously, and they are lacking on the ground situation when we try to collect the samples, particularly for investigation, and even in some of the sensitive cases like forensic investigation, where the animals needs, like a tiger or leopard, needs some specification in the investigation. India do follow some of the guidelines formulated by National Tiger Conservation Authority, and every case of the Schedule 1 species has to be dealt like as unnatural death rather. It should be investigated properly. There are certain guidelines that says that every sample of tiger or leopard should be collected for rabies as well. But when on ground situation we start working, there are many resources which are lacking, which makes the entire investigation and sampling and disease prevalence study very difficult, and rather I would say impossible to get the results on. But again, I mean, when we were talking about some of the disease investigation laboratories, including LACONS, CCMB, forensic labs, regional forensic labs, Wildlife Institute of India, so we came to know that there is large gap between sample collection strategies, the preservatives that are being used for sample testing, the methods of transportation, maintenance of the cold chain, and documentation, proper documentation, verification of the validation of the documents that has to be done. Then when we submit the samples to the lab, that lab gives some reports, and on the basis of that we need to give the interpretation to the concerned authorities, and it has to be analyzed. So as a beginner, as an organization, when we started in 2000, we were facing a lot of problems: where to send the samples, how to send the samples, what should be the standard protocol to send the samples, whether it should be same for every disease investigation. Then gradually we came to know that okay, every situation might be requiring different protocol. So now we have standardized the protocol for dealing mortalities of Schedule 1 species like tigers and leopards, where we perform at least 5 to 6 sample collection for testing, for example, bacterial, viral, parasitological examination. Then we also conduct it for toxicology, and then we also conduct for DNA analysis, where the lab reports has to be validated that this animal was a tiger, whether it was male or female or approximate age, and specification like what landscape that belongs to. So in short, when we execute and design and execute the disease prevalence study, we need to be very, very, very peculiar, very specific about the protocol that we are going to follow. You can see one of the case of langur, which was again rabies positive. Uh, you can see the deformities in antlers and warts in this tiger. So in some cases we can collect the samples very easily if they are brought to transit facility or the treatment facility, but in some of the free ranging wildlife it was really challenging to get the permission to execute the sample collection, to restrain the animal and then collect the sample. So I understand that there are many challenges on ground, but we need to be very specific, and we understand the standard operating procedures for sample collection and its preservation and transportation. When we were talking about vultures, again, I mean, we have been working on vulture conservation for a very long time, and I'm fortunate that we could, save some of the areas where there was a lot of decline in vulture population, but because of support from the forest department, local authorities, and villagers particularly, we could revive the population some of the places. So again, I mean, as a one health practitioner, when we were talking about, understanding the basic biology of the species we are dealing, so vulture is one of the classical example that made us realize that we need to understand the nature and the consequences in different ways. So as we all know that during 90s vulture population was declining a lot, and by 2000 it was almost 99% decline, the dramatic decline ever happened in any single species because of non-steroidal anti-inflammatory drugs that were being used in veterinary practice. So when we talk about the vulture mortality, NSAIDs are the major culprit: diclofenac, aceclofenac, ketoprofen, pneumocillide. These are the major culprits that are being found toxic to vulture in lab as well and in experimental studies. But besides that, the same era was witnessing the upcoming meat industry with globalization, and it caused lack of food availability to the vultures, not just vultures, many other scavenger species as well. There are many reasons that are causing decline in vulture population, including poisoning. But diclofenac was a major culprit when it was found during the studies that it is affecting more than 97, 99% of the population in Southeast Asia. With decline in the vulture population, there was no one at that time when uh, to take place of vultures in wildlife. Unfortunately, feral dog population was also increasing at the same time, and uh, that increased up to certain level that India started witnessing very high rise in rabies cases. So it could be a hypothesis, it could be just a logical thinking that with decline in the vulture population, are we witnessing increase in feral dog population, which results into increased rabies cases. And unfortunately, India is being called as rabies capital of the world, where we are finding more than 80,000 cases per year. So when we talk about vulture poisoning, vulture toxicity rather, then the cascade, if you can see, is something that is affecting one health globally. Southeast Asia is the prime example of this rabies, but I'm sure, and the data is also visible, that many other countries, including Africa, South African continent, those countries are witnessing rabies as well. So my say is that when one species decline, of course, there are many other alternatives that takes place in nature. But if it's happening with a dramatic decline, like in vulture case, nature needs some time. Nature needs some time to cope up with the consequences and take some place and find out the alternatives on long scale of period of time. But when we talk about this cascade, as a one health practitioner, as a veterinarian rather, I would say that there are many challenges when we start implementation of this one health strategy on ground, and how to overcome it. I mean that let us see what can be done, what we need to be, where to start with. So in some of the cases, even in the landscape where we are working on, where we are starting the One Health Center, which has some of the facilities, and we are trying to get it functional with the help of concerned forest department. So how it should be as a veterinary infrastructure facility, what it should include. So it should include treatment facility, operation theaters, animal keeping facility, quarantine areas. If the animal dies, we need to conduct the post-mortem in very scientific and very systematic and serious way. Then the sample collection kit should be available on ground in the center. Then preservative facilitation, then the transportation facilities, the team, the entire team, skilled veterinarians, skilled biologists should be available with this center, and they should have a basic idea about the wildlife health surveillance system, how it should do, how the active surveillance can be done, how the passive surveillance should be done, how the samples are collected, how they are preserved, how they are going to be transported. the data being generated from this all information should be interpreted it, and it should be analyzed, and it should be presented at different level, increasing the chances of collaboration with different institutions, biologists, zoologists, veterinarians, mm, policymakers as well. So basically, when we talk about this implementation, I'm talking about the One Health Center with focus on veterinary facility, but we are also trying to collaborate with some of the institutions where they are trying to collect the samples from the environment. So they are also trying to collect the samples of soil, water, vegetation, and so many other things. So basically, that One Health Center on ground will be very effective in different ways. It will be acting as a disease surveillance model. It will be acting as a treatment facility, sample collection facility for free-ranging animals, and it would be included to dissipate the information to the right candidate, to the right stakeholder, and make the policymakers believe that One Health strategy is very, very, very important for near future. So what should be the way ahead? I mean, how it should be implemented. So some of the experiences where we started detecting the diseases, for example, when we were working in couple of tiger reserves where we were collecting the samples for canine distemper. So the disease was detected in some of the leopards, and the department and the authority was very concerned that whether it's spreading a lot because some domestic counterparts. So we started detecting the disease with collecting the samples from domestic dogs, and now we have collected more than 1,200 samples, out of which almost more than 20 to 25% samples are positive for canine distemper. So when we talk about the host, there are many hosts which are maintenance hosts, which keep the disease spreading. There are spillover hosts which spreads the disease and reproduce the disease to at least one cases with external factors. And there are dead end hosts which do not transmit the disease, but they will carry it for long period till death maybe. So this host has to be identified, and prevention strategies should be focused on that host. So in case of this canine distemper, where we were collecting the samples from some of the villages around the tiger reserves, so we focused that this disease should be detected first. Now the second step was controlling and prevention of that particular disease. So canine distemper. So we started immunization, mass vaccination drive in that area. We are still doing it in Tadobandhari National Park, Tadobandhari Tiger Reserve from Chandrapur district, Maharashtra. So where we are focused on vaccination of almost 3,000 dogs for rabies and canine distemper. But beside that, we started implementation of this wildlife disease risk analysis modeling. So WDRA includes the analysis, collecting the data, analysis of the data which is being, observed by the authorities, and predicting the future outbreaks and the future scenario with number of mortalities, morbidity and mortality. So I understand there are a number of factors which affects the disease at any given time, which includes natality, means birth rate, mortality, means death rate, then immigration and immigration, I mean, to and fro movement of the animals outside and inside this landscape. So wildlife disease risk analysis allows you to predict the extent of the disease, extent of the mortalities, and uh, allow us to focus on, prioritized landscape or the prioritized species in particular season, in particular duration at the particular area. In short, we get a better understanding of spatial and temporal distribution of the disease in terms of prevention strategies. So I understand that vaccination or immunization does not entirely stop the infection, but I'm sure that it definitely reduces the infectiousness of the infectious agent, and it definitely causes some reduction in the disease transmission and the disease spreading up to certain extent. So this is going to be a big strategy. So again, I mean, when we were talking about early warning system like WDRA, we need to be better prepared for vaccination as well. Then as a implementation authority or as a ground worker, I strongly feel that we need to have collaboration with different organizations. At global level, there are World Health Organization, World Organization for Animal Health, ministries, which includes forest ministries and even animal husbandry department, NGOs, and other stakeholders, mainly villagers and the communities living around the protected areas. So what we have started doing in some of the tiger reserves in central India, that we are working with several institutions, include Wildlife Institute of India, the multinational companies, the grantees, CSR funding agencies, the local NGOs and the communities, so that we can implement some of the parameters which are being focused on One Health practice and One Health approach. And now, when the conservation scenario is teaching us a lot, that we need to involve the local communities, the local stakeholder with us. Otherwise, any conservation measure is going to be failure. So, what we were talking about, the awareness and the capacity building of the frontline staff, that includes the forest department and the animal husbandry department and the local NGOs, protecting the habitat, the biosecurity measures that has to be taken, and as discussed earlier, the vaccination, which helps us to control the spread of the disease up to certain extent, to reduce the number of the maintenance host in disease control way. And in some of the cases where the diseases are, found in just like the outbreak, so vaccination may not be the right strategy, but with WDRA modeling, we can definitely predict the outbreak and its extent. So yes, this is what we have been doing, and this is the way ahead. I look forward that as One Health practitioner for disease control, we are starting the same uh, recommendations that are being suggested by WHO and WOH regarding awareness in schools and colleges and the villages, and the capacity building of the frontline staff, forest department, animal husbandry department, NGOs, and the community is happening on ground. And we have reached more than 5,000 people in this year so far. Vaccination, we have almost covered almost 2,000 animals so far. In the past, when we were trying to deal FMD outbreak in Gaur, we had also supported the forest department and animal husbandry department to cover the vaccination of cattle, uh, of 10,000 cattle in the buffer villages of the tiger reserves. So it's a big number that helps us to control the disease and control the outbreak as well. And we are planning to start One Health center at different locations where we what we discussed earlier, we are trying to implement it on ground. I absolutely understand it's very challenging, it's very daunting task, but what we have learned so far, that if we take a step at a time, we can definitely implement One Health strategy or One Health approach on ground as a veterinarian. So that's it. That's it from my side. You may like to contact if you have any question further. You definitely please stay in touch. We would be more happy to collaborate and associate with different institutions to make it happen. And I thank you everyone for your kind attention. I'd like to have questions, if any, please.
Thank you, Dr. Bahar Baviskar, for the wonderful presentation. And I think there are some comments and also some questions for you. Maybe I'll just read it for recording purpose so that it will be.
Please.
There's a trade-off competition between vultures and dogs. As we see increases in dogs at apothears, vultures decline. there are two ways to look at that. Vultures are efficient at eating carrion and regulate dogs. However, once dogs are established, they outcome vultures. This is a comment from Dr. Bipin Prajapati.
Yes, absolutely. That happen in many landscape that because of the dog population, vultures are not getting closer to the carcass. And we have also reported the same thing that there are many dogs that they are trying to, shoo away the vultures, and that's why the vultures are not getting the food. Absolutely, I agree.
Yeah.
It's both way. It's both way.
A question from Dr. Asad. As you working in the field of conservation, what do you think is good if there is breeding in zoo or private wildlife farm breed zoo animals? Will that come in conservation or it's illegal? That's the question. Yeah, anyway, you can.
Okay, okay, I understand. So the basic concern is about ethical consideration. Then certainly keeping the animals in zoo would not be ethical if they are releasable. So most of the time, the breeding centers or the conservation breeding centers are established with an objective that the animals which are captive bred are going to be released back to the wild or back to their natural habitat when the number increases. So conservation breeding centers are something not, it's not just a fantasy or anything like that. It's the need of some of the species. For example, vulture were declining because of the toxicity of non-steroidal anti-inflammatory drugs. Had it not been taken into consideration to, you know complete ban of these NSAIDs or painkillers, we cannot release these captive bred animals and the vultures particularly into its natural habitat unless the threat which caused the decline gets vanished. So we cannot, we are catching the animals, we are keeping those vultures in the breeding centers. It sounds unethical or emotionally it's disturbing. But if we do not so, if we do not do that way, anyway, the vulture as a species is going to extinct. But if we try to breed them in captivity, eliminate the threat from the habitat or the landscape, which cause the decline in that vulture population, for example, painkillers. So now, if the painkillers are not available, they are not visibly used, and uh, there, there are reports that, okay, the ban is effectively implemented, then we can release those breeding centered animals or the captive bred animals and vultures to the habitat. So now they are going to increase their number in the wild. So in that way, I look at it, conservation breeding centers as a, you know, way forward to decline the status of some of the critically endangered animals to uh, endangered species. So we need to improvise our conservation breeding center, which is way expensive and it needs skilled manpower as well. But wherever there is a threatened species, it can definitely help us to bring that animal from extinction to normal status for sure. And I am strongly with the conservation breeding centers in that way.
Thank you, Dr. Bauer, with great appreciation from Nirupama for your presentation. There's a question. Have you seen IBR in Gore?
IBR?
Yeah.
No, so far.
No. Yeah.
There were some of the cases of bovine spongiform encephalitis, but they were not being reported because it's a notifiable disease. So, yeah, I mean, there are certain incidences where we also reported avian influenza in crow, where we found more than 1,000 birds died, but we could not report it because being a notifiable disease, that would have closed the poultry industry around 10-kilometer radius area of that location, which was not economically feasible. So the authorities asked the department that they should not release the report of positive postmortem findings of the bird flu. But I mean, there are many incidences where we could not report some of the diseases, even though they are being found.
Yeah. Thank you. There's one more question from Savani Baviskar. How can we protect urban animals from diseases as common man? Question.
How we can protect?
How we can protect urban animals from diseases from common man point of view.
Okay. So vaccination is the best strategy when we are preventing the disease transmission, as we discussed that reduction of transmission in the maintenance host. That should be the strategy. So yes, vaccination is the best strategy.
Yeah. Thank you. There is one more question, from Dr. Bipin. Are there incidents of FMD reported in Thadia areas in Gors?
Which area?
TADIYA.
Tadia. I don't know Tadia area, but yes, we have reported FMD goes from Navega and Nagzira.
Tadoba. This is wrongly
Okay.
spelled. Now it's
Okay. No. From Tadoba, there were no incidences, but from the surrounding landscape where we were working in 2000 and 2003, there were 8 goats which were affected by FMD. Yes, there are reports of FMD in goats.
yeah.
Not from Tadoba though.
Yeah. Okay. Thank you, Dr. Bahar Baviskar, for the wonderful presentation. And then if there is any more question, I think there is one question, maybe you can answer this also. This is going to be the last one. Is there any protocol to eradicate? I think it's written as eradicate CDV.
Okay. So, eradication, there are 3 strategies of disease dealing. So one is control, second is prevention, and third is eradication. So eradication of any disease has to be very specific long-term strategy for any department, which is almost, I would say, humongous task for any authority. So I would say rather it should be a preventive strategy, and it would be always experimental designing, that it won't be possible for any authority to control and prevent that disease 100%. But yes, we can reduce the chances of spreading the disease by more than 85 to 90%. So I would not say that there is any protocol for eradication of CDV, but yes, there are protocols, and vaccination is one of the most effective protocol for prevention of canine distemper, which is aerosol transmission disease.
Okay.
I could see there is a report of FMD spray to elephants. From my landscape, for sure, we did not have any report of FMD in elephants.
Yeah, that's a question from Arundhati, Sri Lanka. Yeah.
Yeah.
I think that's all for the time being. If there is any questions, maybe we'll just email you and then it'll be just circulated among the participants. Okay.
Thank you.
Thank you, Dr. Bahar, for a wonderful presentation.
Thank you very much. Excuse me. Thank you very much, Dr. Bahar. That was very interesting.